there are four main types of ribonucleotides present in the cells, and it depends upon the types of nitrogenous bases present in a ribonucleotide. You know the structure of a ribonucleotide. There are three components. One is pentose sugar, ribose sugar, the other one is phosphoric acid, and the third one is nitrogenous base. The two components, that is phosphoric acid and a ribose sugar, they are same in all the ribonucleotides, and the only difference is in the nitrogenous base. So as you know, there are four types of nitrogenous bases present in a ribonucleotide. One nitrogenous base is present in one ribonucleotide, but overall there are four types of ribonucleotides. So there are four types of nucleotides which are present in a ribonucleic acid. These four types, they depend on the type of nitrogenous base. As you can see, this colored portion is a nucleoside. You know, this is a nucleoside because a pentose sugar and a nitrogenous base, when they both combine, they make a nucleoside. And here the nitrogenous base is adenine. So this nucleoside is adenosine. And when to this adenosine a phosphoric acid is attached, it becomes a nucleotide. So this adenosine becomes adenylate. Let's move to the second type of ribonucleotide present in the nucleic acids. Here it is guanine. So in this colored portion, a guanine joins with a pentose sugar and they make a guanosine. And when to this guanosine a phosphoric acid is attached, it make a guanylate. So next, here are two other types of ribonucleotides. Again, the colored portion, this colored portion is a uridine because the nitrogenous base present here, this is uracil. So to this uridine, one phosphoric acid group is attached and it becomes uridylate. This is third type of ribonucleotide present in the nucleic acids. The fourth and the last type of ribonucleotide is cytidylate. In a cytidylate, the nitrogenous base is cytosine, and this pentose sugar and this phosphoric acid is present. So when a cytosine attaches with pentose sugar, it makes cytidine. And when to this cytidine a phosphoric acid is attached, a complete nucleotide, that is cytidylate, is formed. So these four types of ribonucleotides are present in the ribonucleic acids. Next, how these ribonucleotides are joined together, because I said ribonucleic acid is a polymeric compound. So many ribonucleotides join together to make a molecule. So here you can see this is one ribonucleotide and this is second ribonucleotide. So in this ribonucleotide, one phosphoric acid is attached to its 5 carbon, and this attachment is through ester linkage. So the hydroxyl group present here in this phosphoric acid, it attacks to the hydroxyl group present on the pentose of next ribonucleotide. So in this way, a condensation reaction takes place here. A water molecule is removed, and here you can see these two ribonucleotides, they join together and they make a dinucleotide, in which these two nucleotides are linked together through phosphodiester linkage. Why this is called phosphodiester linkage? Because Here one phosphate group is present, and this phosphate group, it is linked with one pentose with a ester linkage, and the second ester linkage is present here. So whole this is one phosphate and two esters, so it is called phosphodiester. So in this way, a polymeric compound consisting of many ribonucleotides are formed. So in this way, many ribonucleotides, they join together and they make a polymeric chain, which is called a polyribonucleotide or a ribonucleic acid.